Hi all, Dr. Clark here. Today for General Biology lecture series, I'm going to talk about the 4 unifying theories that really no matter what topic we are discussing in biology, these 4 theories will have some input or have some place in whatever we're discussing. Okay? These theories, again, remember we were talking about hypothesis and theories last lecture. Remember that scientific theories are, have a high degree of certainty to them, meaning that hundreds of experiments have been done on that original hypothesis or the original hypotheses. So it could be multiple hypotheses. And from that, there's so much data and so much evidence for that hypothesis being a true explanation for that natural phenomenon, that they've been elevated from hypothesis to theory. Okay? And so that's what a theory is to a scientist. Remember, we already talked about to the general public, a theory is a guess. These are not guesses. Okay? These are well explained hypotheses with lots of data, and they are the accepted explanation for that natural phenomenon. So let's get into it. Okay? So what are the 4 unifying theories of biology? First, the cell theory. Okay. And we'll go through each one of these and some of the individuals that were players in each one of these theories and what each theory, the main tenets to each theory. Okay. The gene theory, the theory of heredity, and the theory of evolution. Okay. These 4 theories, no matter what subject we're talking about in biology, these 4 theories have some skin in the game, if you will. Okay? So they all will have a piece of whatever we're talking about, whether we're talking about plants and plant structures and photosynthesis, or we're talking about water quality, or we're talking about how cells go through osmosis and diffusion. Every single one of these theories play an integral part in explaining those processes. Okay, so first, the cell theory. The cell theory, or how life is organized. Okay, we kind of already talked about this a little bit. Okay, well, it starts really with Robert Hooke. Okay, Robert Hooke in 1665 was, and at the time, what you would consider a leading biologist or a leading scientist. Okay? Robert Hooke was also, I guess you could say, an amateur engineer, in that he realized that by stacking glass, you can make a magnified image. Okay? He wasn't the first to make a microscope, okay? And we had telescopes already. Okay? But he was the first to ever look at life under a microscope. So he's really the first to use a microscope to examine living tissue. Okay, at least on record he was. There's a lot of other information about Hooke, and I'll bring him back in when we talk about cells, and I'll tell a lot more stories about Hooke. He was a integral biologist. He was very hated at times, though. Newton hated Hooke, and part of that might have been that Hooke had, prior to Newton, Hooke had already wrote about gravity. Okay? I know it's a big spoiler because everybody's like, oh, Newton, the gravity dude. Hooke had already wrote about it prior to Newton. And in some people's eyes, Newton stole Hooke's information. But regardless of that, even if that's not true, okay, Hooke was a well-known scientist. He had a big ego. He liked to get his hands in every cookie jar. So he was working in cell biology, physiology, physics, chemistry. He was working in all kinds of things. Okay? And a lot of people were heard about that. A lot of scientists were heard about that because Hooke always had an opinion about everyone's research. Okay? But nonetheless, he discovered cells. Okay? And he did so by creating a microscope and looking at cork, okay, which is, for those of you who don't know, I mean, most people know what a cork is. Like, you know, it stops the wine, or it plugs the hole of the wine bottle. That's cork, okay? But what cork actually is, true cork, is it's right underneath the bark of a tree. You'll have this holy material, which is a protected material of dead tree cells, and that's cork. Right? Well, Robert Hooke discovered cells by looking at cork, okay? And when he did so, he saw squares or rectangles. Okay? And some stories say that, you know, the cell or the rectangle made him think of jail cells. Okay? But most of the stories suggest that he actually, it reminded him of the rooms that monks used to stay in, and they're called cells, and so are jail cells, but they're called cells, and that's where the name came from. It's from monk inhabitants. Okay. Next, you know, a little bit later, Antoine van Leeuwenhoek, he was a much better engineer, made, you know, hundreds, over 250 different types of microscopes, okay, and utilized them. Okay, so he took what Hooke had done and made it better. Okay, and in doing so, he discovered single cell life. Okay, so he's looking at water and stuff like that, and discovered, you know, single cell protista. He was probably most likely, although, you know, in the 1600s, the note taking and what they called things were different than it is today, but he most likely also saw bacteria. Okay? So Antoine van Leeuwenhoek, okay, he was kind of one of the precursors to the cell theory. So Robert Hooke's first, and then Leeuwenhoek, and then, okay, Schleiden and Schwann, okay, these guys, you know, almost 200 years after Hooke and Leeuwenhoek had made microscopes, these guys used microscopes. They didn't make microscopes, but they used microscopes to describe life. Okay, and I don't want to take anything away from them because they described lots of life. Okay, Schleiden was a plant guy and Schwann was an animal guy, and they looked at everything. Okay, and they came up with one of the tenets of the cell theory, and that is all organisms, all living organisms, are comprised of cells. Okay? And that cells are the basic unit of life. So these are the first two tenets of the cell theory. There are three pieces to the cell theory. Okay? First one, all living organisms are comprised of cells. Okay? Cells are the basic unit of life, meaning that you can have single-celled life. Okay? And then the third tenet came later. Okay? Now, it depends on who you want to cite for the third tenet and the role that other biologists play, etc. Okay, I think credit needs to go to one man. Okay, and we'll talk about him in just a second. I have a picture of him in just a second. But in the third tenet was all cells come from pre-existing cells, or all cells come from other cells. So you got to remember, even in the 1800s, during Darwin's time, okay, so all this cell theory was worked out before, most of it was worked out before Darwin published The Origins of Species. Okay, but even to that point, all the way up to Darwin's time, and a little bit after Darwin's time, most of the world believed that a higher being created cells just poof into existence. So in other words, things like getting sick. So if you were an individual and you were sick, you were ill, okay, during the 1800s and prior to that, okay, it was thought that, well, you're ill because you did bad things. Okay, so maybe you sinned, and a higher being, God, a god of some sort. Okay, there's lots of different gods and lots of different religions, etc. But a lot of them believed this for a long time, that God made you sick. Okay, it's that entity is what made you sick. Okay, and it wasn't until this man right here, okay, whose name is Louis Pasteur, it wasn't until he started doing experiments that third tenet, that all cells come from pre-existing cells, came about. Okay, so we had a cell theory. Again, like I said before, theories are tentative. Okay? You can add to them, you can delete things as more information comes. That's why, biology is always changing. That's why nothing is of complete certainty, and why in biology we don't prove anything. Okay? It's because it will change with more information. Okay? And that's what happened to the cell theory. Okay? The third tenet was added after Louis Pasteur did his pasteurization work. So one of the classic studies is shown here, what Louis Pasteur was doing. So he would take what we call nutrient broth. Okay? So it's basically water with nutrients in it. Okay? And these nutrients are needed for things like bacteria and fungi to grow. Okay? And he would take them and he'd boil them, sterilize them, so nothing was growing. Okay? And then he created this glassware, which we call the swan neck, or sometimes it's called Pasteur glassware. Okay? So he created these swan neck glasses. Okay? And some of them had removable necks, but some of them he'd just break the neck off. Okay? It just depended on what time period you're looking at his experiment. So he'd fill the flasks, with this nutrient broth. Okay? He'd boil it up, and then to some he would leave the swan neck, and to others he would break the swan neck off. Okay? And then he'd sit them in the room, and he'd wait for something to occur. Okay? And it wouldn't be but a few days, and the ones that had broken tops would become cloudy and have bacteria and fungi in them. Okay? But the ones with the swan necks would stay clear and be void of any living organisms. Okay? So Louis Pasteur, over many years of doing this experiment over and over and over again, came up with the conclusion that a higher being is not creating cells and putting them in these flasks, that the cells are actually in the air floating around, and that when they have access via a broken top to this nutrient, and they go in, because the air can travel through here, they go in, they have the nutrient, they reproduce, and the nutrient broth has a lot of organisms in it. In the swan neck case, the air is not going to fight gravity, okay? So there's no way for the bacteria and fungi spores or cells to get in here, and they stay clear forever for the entire experiment. And so this is where Pasteur came up with all cells come from pre-existing cells. Okay? Now that's not the only thing that Louis Pasteur did, and we'll talk about him many, many more times. Okay? When we talk about pasteurization, when we talk about germ theory, when we talk about theory of disease. Okay? Louis Pasteur was very prominent in culturing bacteria and things like that, and probably viruses. Okay? He didn't know there were viruses at the time, but his work was done on both bacteria and viruses, and looking at disease transmission, etc. Okay, but nonetheless, the third tenet, okay, of the cell theory is all cells come from pre-existing cells. Okay, the gene theory, or the molecular base of inheritance. So the gene theory really came about from the discovery of DNA. Okay, so up to the point of really the 1940s, okay, we didn't know. We knew that DNA existed in the 40s, because we knew about chromosomes in the 20s, and maybe even before that. Okay, but in the 1940s, there was enough information that researchers could start putting together what DNA consisted of. Okay? What was its making? What was its shape? And then once you can figure out what it's made of and what its shape is, then you can figure out things like how does it reproduce? Okay, what is it coding for? These kind of things. Okay? And a little bit later, outside of the 1940s, after Watson and Crick and Wilkins and Franklin's research on DNA structure, you start to see that pieces are beginning to be put together. Okay, there's the race for the double helix is over. We figured out what shape DNA was, or is, sorry, and now we can start putting things together. Like, what does DNA do? How can it change? These things. Okay? And it was discovered that genes or DNA codes for proteins. Okay? But proteins, the only way you can make a protein from a DNA code, okay, is by switching the DNA code into RNA. So you have to think of it kind of like you're reading an instruction manual. Okay? But the instruction manual is in Spanish. Okay? So you have to translate it from, and maybe you don't speak Spanish, okay? Or maybe you do speak Spanish, but not very well. Okay? And you have to translate it from Spanish to English, and then English into building whatever it is that you're going to build. That's the exact same kind of process that DNA has to go through. DNA has to be translated into RNA, or transcribed, rewritten into RNA, okay, and then translated from the RNA into a protein. Okay? We'll come back to this. I guarantee we'll spend lots of time if that was confusing to you. Okay? So proteins, okay, are the building blocks of all organisms. Every single organism on the planet that's living is built of proteins. And those proteins are coded by their DNA, which is rewritten as an RNA code. Okay? There are a few organisms where the DNA doesn't exist, and it's just the RNA code. Okay? And we'll talk a little bit about how that works. Okay? But nonetheless, proteins are what builds everything that you can think of. Okay? Your hair, eye color, okay? Whether or not you can roll your tongue, okay? The shape of your fingers, whether or not you have hair on your knuckles, okay? The shape of your arms, okay? Your bone mass. Okay? All these things are the result of... Okay, so gene theory. Alright, so it really started out with that discovery of DNA, the structure of DNA, how it is transcribed into RNA and then translated into proteins. And from there, we actually can figure out lots of different things. We can figure out where the DNA is housed, okay, where genes are housed, okay, how many genes we have on each chromosome, how many genes we have collectively. That was the Human Genome Project, which was finished in the 90s. Okay, and then from there we can see, well, why is it that certain cells, okay, they all have the same DNA, they all carry every single gene, but why do they look different? Why do intestinal cells differ from nerve cells, from muscle cells? Why is it that your hair looks different than your fingernails, which looks different than your skin, etc., etc.? And that really came about from gene theory. And I'll tell you that this is research that's ongoing, okay, and is probably one of, if not the field that most people are interested in currently in modern biology, and that is human genes. Okay, because the more we can understand about human genes, the more we can understand about diseases, malformations, okay, the differences between individuals, what happens when your genes shut down, what's the tie to cancer and genetics, okay, and just a ton of different things that can come about from studying gene theory. Okay, the theory of heredity, okay, so this really was Mendel's work, okay, and this is, you know, how genes are passed from one generation to the next. Now, I have to talk about this, and we'll come back and talk a lot more about Mendel. Okay, Gregor Mendel was the individual who really, spearheaded and is known as the father of genetics, but spearheaded the theory of heredity. Now, when he did this work, there was no such thing as genes. Okay, so he didn't actually know. Mendel didn't actually know that there was such thing as DNA, chromosomes, alleles, loci. He didn't know anything about that. He just knew mathematically it worked. Okay? And so Mendel's theory of heredity really gave rise to the field of genetics. Okay? And so between gene theory and the theory of heredity, those two theories are, I would probably say, 75 to 80% of all biological papers and biological information that's going on today are directly related to these two theories. Okay? Understanding genetics, understanding diseases based on genetics, etc. Okay? And, there's other things that were built into this. So Mendelian theory, so he has two main theories that come along with this. Okay, the theory of segregation, and then the theory of independent assortment, okay, or chromosomal theory. Okay, they're built into the theory of heredity. Okay? And so it really came about from Mendel's work on peas, Mendel's work with mathematics, okay, and later confirmed. Okay, so this is the cool piece. Mendel was doing this work in the 1850s, okay, when we didn't know anything about DNA, chromosomes, segregation, didn't know about, mitosis and meiosis, and we'll come to all this if you don't know what that means. Okay? He didn't know any of that. But the cool thing is 50, in some cases 100, 150 years later, we're confirming with modern technology the ability to segregate genes now. At first it was chromosomes, and now it's genes and segments of genes and things like that. We're able to confirm that what Mendel was talking about in the 1850s and what he figured out mathematically, for the most part, is true. It's still valid. All right, so there are a couple books, that I like to share, books that I found helped me with subject matter, or were very interesting when it came to these subject matters. Okay, one is The Monk in the Garden. So Gregor Mendel was a monk who studied peas. Okay, and this book is about his work, about his writings, about the loss of kind of all of his genetic research, and then how it was found, roughly 50 years later. Okay, so excellent book, by the way. Link is below. And then Rosalind Franklin, okay, sometimes known as the Dark Lady of DNA. Most people, when we talk about DNA and the structure of DNA, if I asked students, they heard about Watson and Crick. Okay, James Watson, Francis Crick. That's who discovered structure of DNA. Well, in actuality, Rosalind Franklin probably discovered the structure of DNA. Okay? She was the first person to write in a journal that she thought the structure of DNA was double helix. She was definitely the first person to note that it was helix, and she did in an open forum. But then she was most likely the first person to write that it was a double helix. Okay? Rosalind Franklin would have won the Nobel Prize along with Francis Crick, James Watson, and Maurice Wilkins for the structure of DNA. But she died, and you cannot win the Nobel Prize if you're dead. So they were giving her credit, but most of the time she's kind of the forgotten piece of the double helix. And it was her X-ray crystallography work that was by far the most important to the piece, to the discovery of DNA. And in my pure opinion, James Watson and Francis Crick would have never figured it out, not even close, without Rosalind Franklin. Okay, excellent book talking about her life, her research, and just a very good book about another lady of science that often is forgotten. And I'll bring up a lot of females in science that just don't get the credit that they should, because to tell you the truth, it's beginning to change, but it's still a man's world in most aspects, especially in science. Men far outnumber female scientists, and they did so, in the 1800s and in the 1900s. Okay, and they are still in the 2000s. Okay, so that brings me to the last theory. Okay, and that is the theory of evolution. We've talked a little bit about it. Okay, but Charles Darwin is kind of the father of evolution, but he's not the only player in evolution. Okay, we'll talk about a lot of players in evolution. Okay, we'll talk about Alfred Russel Wallace, and Mendel was a player in evolution. There's lots of individuals that are players and very important for the theory of evolution. Okay, but Charles Darwin wrote something that's probably misrepresented, or the words have many different meanings to some people, and he wrote descent with modification. Okay? And this term, descent with modification, he wrote it to explain the diversity of life. Okay? In that organisms all came from a common ancestor. Okay? And that as life progressed and as the climate or the environment changed, organisms evolved. Okay? And they did so by very minute changes often. Okay? And that an organism would produce offspring, none of them being identical often, okay, depending on what kind of reproduction it was. But most of the time, if we're talking about sexual reproduction, the offspring are not identical to their parents. They have minute changes, okay? And some of those changes make that offspring better suited for the current environment. And that's where the descent with modification comes from. If you descend enough times and you have enough modifications, you go from one species to another species. And that's kind of the nuts and bolts of Charles Darwin's theory of evolution. Well, the cool thing about this, just like Mendel's research in the theory of heredity and gene theory, even Mendel has his fingers in gene theory also, really came about, that these individuals in the 1850s were discovering things. And they were making hypotheses and testing hypotheses and coming up with theories before genetics was even around. So they're making hypotheses about how things change over time, how genes change over time, without knowing that there's even such thing as genes. Okay? And so this is amazing from most aspects of biology. When we got modern genetic techniques, we could test to see if Charles Darwin was wrong. When he said that descent with modification shows our ancestry to chimpanzees, modern genetics allowed us to test that. We could take the DNA from humans, and we could take the DNA from chimpanzees. We could plot them all out, and we could look at the differences. And if it's true that we share a common ancestor with chimpanzees and that they're our closest ancestor living today, then they should be closest based on differences of DNA. And that's exactly what happened. Okay? We share 98.1% of our DNA with chimpanzees. Okay? Next in line are gorillas. Okay? We share about 96% or so. There's about a 4% difference between humans and gorillas. Okay? And on. We can start looking at all of them. Orangutans. And then we can go into monkeys and old world monkeys, new world monkeys. Then you can just keep going back. Okay? And that's what genetics allowed us to do, is test the theory of evolution. It allowed us to test Darwin's theory that he put forth 100, 150 years prior to see if he was right. And nonetheless, he was. So, for example, we look at the beaks of finches. Okay? Darwin suggested that all the finches that were on the Galapagos Islands, all the different forms of finches, okay, they all came from an ancestral finch that lived on the mainland in Ecuador. Okay? And somehow it got blown out to these islands. A few individuals, okay, a handful of individuals, maybe a couple times, got blown out to these Galapagos Islands. When they were there, they either adapted or they died. So their offspring were either adapted for the environment or they all died. And, it was a great hypothesis. But there was really no hardcore way that you could test it until today. We now know that we can take ancestral finches, we can take finches from the mainland, and we can look at their DNA. And we can look at the DNA of the finches that are on the different islands. And we can isolate what gene might be different. And we did so. Okay? It's the BMP4 gene, okay, that seems to be what's causing finches to have different beak shapes. Okay? And that's what Darwin was noticing, is that the beak shape was different. Now, overall, the birds were very, very similar to each other, but their bills or their beaks were different, and they were modified for different food resources. Okay? And so now we know that there is a gene, a BMP4 gene, okay, that differs between every one of these organisms. Okay? How much is expressed, when it's expressed, these kind of things differ, and it will cause a different protein base to their bill. Okay? So you have a lot more BMP4 gene turned on, you have a bigger, thicker bill. Okay? A lot less, okay, a thinner bill. Okay? And so modern genetics, along with evolution, along with heredity, and along with cell theory, okay, have really allowed us to kind of examine biology, no matter what biology we're talking about, whether we're talking about ecology, physiology, okay, cell biology, germs, okay, whether we're talking photosynthesis, whatever it is, it doesn't really matter. If it's living, okay, and it's a living system, those four theories allow us to examine them. Okay? All right, so next time we'll go into chemistry.